Hi, my name is Josephine Robb, and I'm going to be presenting to you about the ABP subpopulations involved in infant-directed aggression. So, parenting is incredibly stressful, both in the human world and the animal kingdom. But in the animal kingdom, when a parent is placed under stress, this can lead them to adopt maladaptive parental behaviors, such as being neglectful or even infanticidal toward their offspring. So my project is looking at the neurological basis of these changes in behaviors and seeing what may cause that change in behavior. To study that, I looked at the PVN, or the paraventricular nucleus, which is an area of the brain, according to Auction 2019, that has been linked to areas that are already active during infant-directed aggression. Additionally, it has been found, according to Schwartz in 2010, that it's involved in the stress response. There are certain cells in the PVN called ADP cells that play a large role in many social behaviors, but these social behaviors are very diverse. We have aggressive behaviors as well as parental behaviors, which led my mentor and I to believe that there must be subpopulations within these ADP cells. And specifically, I chose to look at protonorphine and CRH. And this is because they have both been linked to the stress response, and Peterson in 1991 found that there is a strong correlation between CRH concentrations and aggression towards an offspring. So my thought process was essentially that these aggressive individuals their stress response must be triggered, and therefore they're interpreting the pup as a stressor or a threat to them, and that's why they react aggressively, whereas other individuals may react more parental. So the purpose of the study is to examine the ABP subpopulations found within the PVN, specifically protonorphine and CRH, and the hypotheses are that ABP neurons of aggressive individuals will express more protonorphine and CRH, and that these neurons are more likely to be active. To study this, first we had to test behavior, then we did the tissue testing, and then finally analysis. So for behavior, we use mus musculus because they have a very similar distribution of chemicals as humans do, and we use 4 virgin males, 4 virgin females, and 4 fathers. Virgin male mice are instinctively infanticidal when exposed to a pup, whereas virgin females are instinctively parental. And what's interesting is that after the gestation period and after copulation, fathers, even if it's not their pup, will be instinctively parental towards an offspring. So we did a behavior test, and we found that all 4 of those individuals within each group corresponded to their typical behavior. So all 4 virgin males were instinctively infanticidal, virgin females parental, and fathers parental as well. After my mentor harvested the tissue, I then started tissue analysis using RNA-scope treatment, which is a type of treatment that uses these little molecules called probes that detects mRNA within a cell. And as you know, mRNA is very specific. So if that mRNA is found within a cell, it's only producing that specific peptide that is the bases correspond to. Then we imaged the tissue, and finally we used various different analysis software such as ACD Bio, Fiji, MATLAB, and then finally we conducted a Fisher-Exact test to see if our results were significant. When looking here from left to right, we have the coronal cross-sections of a virgin female, a virgin male, and a father. And when we look at the virgin female, there seems to be a very even distribution of different subpopulations demonstrated by different colors. Again, I specifically looked at protonorphine and CRH, so that is found. CRH is in magenta, protonorphine is in red, ADP is in orange, and CFOS is in cyan. And what CFOS is, it indicates when a neuron has been active during behavior. So here we see a very even distribution, but when we look over to virgin males, we see a high concentration of orange cells, or AVP cells. And then when we look to father, there seems to be an even starker contrast between both virgin males and fathers, where they seem to have very little AVP cells to begin with. When it came to quantification of the colocalization, meaning that they express both ADP and protonorphine, we found that yes, fathers had significantly less AVP protonorphine cells when compared to both virgin males and virgin females, and the same trend was found during the activation of these neurons. So they had less of those neurons, and less of those neurons were active when compared to virgin males and virgin females. An interesting result was that there was no significant difference found between virgin males and virgin females, which could indicate that yes, protonorphine may trigger the stress response, but the stress response looks different across the sexes. So whereas a female, if their stress response is triggered, they're more likely to be parental, a virgin male is more likely to be aggressive, and that's why we see the discrepancy in behavior. We next look at the colocalization of AVP with CRH, and again we found that fathers had significantly less AVP-CRH neurons when compared to both virgin males and virgin females. However, in this case, there was a significant difference found between virgin males and virgin females, which could indicate that CRH has a much more universal role in dictating the behavior of mice towards offspring, which is supported by Peterson in 1991, where they found that they are more likely to be aggressive with more CRH. When it came to the activation of CRH neurons, we did not find any significance in the results. However, there was a trend towards lower activation in fathers and much higher activation in virgin males. So overall, virgin males had a higher number of these ABP subpopulation cells and a higher number of those cells that were activated when compared to fathers, which has the big discrepancy in behavior. So this could explain that perhaps because they have a higher number of these cells and they're more activated during behavior, this could explain why they act so aggressively towards an offspring, whereas a father is more likely to be parental towards an offspring. This was a relatively small study because it was a pilot study, so in the future we hope to do this with more, so that way we can see if the results with the activation of CRH neuron, if they're actually significant, if there is a trend there. There is no direct link, but we can speculate that human infant-directed behavior may be modulated by some of these chemicals, because we do see maladaptive human behavior such as postpartum depression, which could mean that there's an imbalance of some of these chemicals. In the future, I hope to look at the other ABP subpopulations, such as preproenkephaline, as well as examine ABP mRNA, which is a much more specific type of analysis. Thank you very much.